Second. Okay, perfect. Hello, everyone. Welcome to this week's CGIL seminar. Today, we're delighted to have Vera van der Heijden, who completed her bachelor's degree in animal science at Wageningen University in 2014, and shortly after began her master's at Wageningen, specializing in animal breeding and genetics, quantitative genetics, and terrestrial animals. In December 2016, Vera started working at Hendrix Genetics Aquaculture as a geneticist. During the years that followed, Vera specialized in Atlantic and coho salmon breeding, but also has experience working with rainbow trout and white leg shrimp. Vera mainly focuses on data processing, development of new traits, and selection of candidates. And with that, Vera, I'll pass it over to you.
Yes, thank you very much. First of all, thank you very much for the invitation to be a speaker here at your seminar. I'm very flattered with the invitation. And thanks everyone for listening to me today. So I will be telling you a little bit about the differences between aquaculture breeding and terrestrial animal breeding. Um, I've been working on terrestrial animals during my studies and only switched to aquaculture after. And I have to say, I was quite surprised by some of the specifics of aquaculture breeding that there are. And I'd like to share some of those with you. First, to give you a little bit of background about Hendrix Genetics. Why is this not working? There it is. We are a multi-species company, and we believe that the world can be a better place if we improve the genetics of our animals. So that means to improve their sustainability, to improve their welfare, and to make everything a better place, a better world for the next generation. We operate worldwide in all the countries that are green colored here. That's where we have actual operating parts of our company. Like I said, multi-species, you can see the species on the left. Well, it's a very big multinational global company. This is the brands that we have, genetic brands. You may have heard of some of them. I'm not going to name all of them because I don't think that that's very interesting right now. What I would like to talk about is the Hendrix Genetics aquaculture part. We have fish in 4 countries, yes, 5. We have fish in Scotland, that's our land catch line. We also have this line in Chile. As you see, there are some beautiful pictures that you can take around the farms. We have trout in the USA, near Seattle, that is. And we have white leg shrimp in Hawaii and also in Ecuador. So that's the 4 brands that we have. We have land catch, the Atlantic salmon, we have trout, and we have white leg shrimp. So now about aquaculture breeding and the differences that we have with terrestrial animals. I've tried to keep this a little bit global for all aquaculture species. Sometimes there's some specifics for the species that we have, but in general, you can say that this is about species that are kept for production. So it's not about the nice aquarium species that you will have at home. It's not about the koi, but it's about the species that are really kept for production, where we have also indeed completed the life cycle, because that's not even the case for all species. And then this is an equation that you all know. To improve the genetics means to have selection intensity, that's the I that we have, means to have accuracy of selection, the R, means to have the genetic variation, and it means to have the generation interval. And if you combine that information, you will have the genetic improvement that you can gain throughout the years. And we have in aquaculture some advantages there that you will probably be amazed of. And then in the next few slides that I'll be showing, those are really about this equation, and you will see the equation on the top right part of the slide. First of all, the numbers that we have. This is where it gets funny. Trout, we have 5,000 eggs per spawn, so that is one family of 5,000 individuals. Atlantic salmon, 15,000 eggs, every female. And then shrimp, we have 70,000 eggs per spawn. Trout and Atlantic salmon usually spawn only once during their lifetime. Shrimp can spawn every few days for 3 months in a row. That is a lot of animals. So if you're talking numbers, if you're talking selection intensity, the answer is yes, we do. That's a high selection pressure that we can have. And we try to make the most of that. We actually have to restrict the number of animals that we can keep during for one family, because 15,000 animals per family is just too much in terms of what you can have in biocapacity. We simply cannot keep those numbers on the farms that we have. Then moving on, something else that we have, and this is specifically for salmonids. We have a very interesting family structure where we can have a very clean full and half sip family structure. So that means that we can really choose which male to mate to which female. And we will often choose to mate males to multiple females and sometimes females to multiple males. We have more than enough eggs for that. And that makes a real clear full and half sip structure that we can actually use to increase this accuracy of selection that you see in the equation. So to visualize like this, now this is not exactly the family structure that I would like to see, but it is quite clear. This is as simple as our family structure can get. So just really like male, female, this to that. And it doesn't get confusing with one female to 5 different males and one male to 10 different females, etc., etc. It's a very clean structure that we have. That has two big advantages. The first is that it's perfect for the breeding scheme. If you're talking about accuracy of selection, we get a lot of information coming from full sips, coming from half sips, coming from cousins, coming from everywhere. Think of those numbers that we have per family. It's amazing. It also has the advantage that we can use very cheap, low density chips when talking about genomics. That's not a topic that I will be discussing in depth because there's not that much differences between terrestrial species and aquaculture species, but this is one. So genotyping per animal can actually be done relatively cheap. That's also needed because the value of one breeder fish, one nucleus fish, is actually just not as high as it would be for terrestrial animals. Another advantage that we have is the heritabilities and the variation that we have. Recent domestication, fish have only been domesticated for the past decades. I think it's 50 years for trout. And that's the longest. So no, 70 years, I'm sorry, and 50 years for Atlantic salmon, and a lot of other species only even more recent. That means that we have a very high genetic variation among the stock, and it also causes the high heritabilities that we have. So, for example, the harvest weight or slaughter weight, as you would probably call it, 0.4, which might be a number that you would be amazed to hear. Maybe not, I don't really know, but at least for us, it's a very convenient, very nice number. So we can make good progress every year and every generation. Disease resistance would be 0.15 to 0.5. It depends a little bit on the disease and on the species that you're talking about, what it actually is. But in general, it means that we can make good progress, including the nice variation that we have. There simply is a lot of difference between families, and we can really, really make good progress. If you're talking harvest weight for Atlantic salmon, that's the species that I mostly work with. Say they are 2.5 kilograms at harvest. We would say that we have about 0.5 kilogram every generation added to that. So that's huge increases. And that doesn't always actually translate into that 0.5 kilogram because they can also choose to harvest the animals sooner. So just to have them shorter in the water, which has other advantages that I will come back to. So that's the accuracy and also the genetic variation in the equation on top. And then one last addition to that is this one, the generation time that we have. Generation time in Atlantic species can be very, very short. This maybe some of you know about insects and their generation time. This is not too different to that. So talk about shrimp, 6 months, tilapia, 9 months, African catfish, 1 year. Then we have trout and coho salmon with 2 years, and Atlantic salmon is quite long actually. It's 4 years generation interval. So you see that for like shrimp and for tilapia, and also for the catfish, it's amazing what you can do. Shrimp just two generations in 1 year. Their progress just goes through the roof. It's really amazing. So that's the kind of advantages that we have really with regards to this equation: the intensity, the accuracy, the genetic variation, and the generation interval. And that's some of the things that I was really amazed at first to notice, like, wow, this is really different from terrestrial animals. Then there's some other advantages that are more subtle, but I think they are still very, very useful. And I've summarized them here. I don't have nice, clean clean icons to illustrate them, but I got some pictures for you. First, on the top right, we have development and maturity control. So the advantage of most fish being actually cold-blooded species is that we can control their development using water temperature, especially in early life. So we have a spawning window that will last for like a few months, 2 to 3, sometimes 4. It all depends on what you want, basically, because we can also control that. But your nucleus animals, your breeder animals, you want them close together so that they will also be mature close together. And so that they will be at the same stage of development at the time that you will be measuring those animals. And to do that, you can work with the water temperature to actually speed up the later fertilized eggs and slow down the first fertilized eggs and actually bring them together. It actually goes so far that we don't measure development in days or in age of the eggs. We measure it in degree days. So that means the average temperature per day that the animal have had, and then we sum all of that. So if it's been kept for like 7 days at 8 degrees, that's like 49-degree days. That's actually the age, the way that development is measured for those eggs. And then if you talk about maturity control, we can also do that. And then we can work with both the temperature and also the light regime. So what you see here in this picture, there's actually a tank where you can keep, well, depending on the size, a few hundred to a few thousand animals in there. Atlantic salmon tank this is, so that would be fish. And this tank is completely covered, now opened up but normally closed, with some darkening material so that it doesn't actually let through any of the light from outside. So if you close those doors and there's no light inside, then it will be completely dark. And then you can use the lights inside to actually mimic the season outside. Light in combination with the water temperature. So you can actually speed up or slow down the fish's development in terms of maturity by pretending it's a different season out there. So you can like make summer last a bit longer or make winter last a bit shorter. And by doing that, in Atlantic salmon, you can like make the animals mature 1 year early or 1 year late, depending on what you want. So in terms of 4-year generation interval, normally that would be 25% earlier or later. So that's quite a lot actually that you can you steer that. I don't know, maybe you can make it even quicker, but I don't think we've ever tried that. Then bottom right, there is less emotions involved. Farmers, fish farmers, and aquatic species farmers, they will not have a favorite animal. They will not like a certain color. They won't have any behavior that they want to see. The fish, they don't specifically have to have any features. They have to be healthy and they have to grow well. And that's what matters to the farmers. So what that means is there's no attachment to certain animals that they would like to use more often. There's no traits that are not economically worthwhile that we still have to select on because it's what the farmer wants to see. We can focus purely on the genetic aspect of fish farming, of aquaculture breeding. And I think that's quite an advantage actually, because you don't have to work on color and you don't have to deselect a specific animal because I don't know, it moves a bit more than the farmer wants or something like that. So you can really just focus on the traits that have an economic value. Then bottom left, I'm sorry, we have fertilization out of female. This is an this is in most aquatic species, and it's an advantage in some monads mostly. Because we have total control of the fertilization process. In shrimp, it's actually so that the female will keep the eggs close to her, and she will be there to select the males that will fertilize those eggs. So we don't have control there. In some monads, it's actually the case that the female will be stripped, as we call it. So that means to take out the eggs of the female. I will show a picture in the next slide where you can see this happening actually. So the eggs are outside the female. They are literally in the bucket. You can actually see it's a zoomed in picture, but this is actually, they're in the bucket there. And we will add the sperm or the milt, as we call it, from the male, which is also outside of the body. It's been collected and we just have it in a simple flask. And we will add that to the eggs and we will stir them around, and we will have, in case of Atlantic salmon, 15,000 animals created. Just like that. It all happens in a few minutes. So it's really amazing once you've seen it happening the first time. And well, that means that's just total control over everything. We can decide to split the eggs in 2 or in 3 or in 5 or however many families you want to create from this one female. And you can all keep them separate, and you have got total control over that. That also means, and that's for all species, because that's also the case in shrimp actually, there's no gestation period that you have to take into account. Once the animals have spawned and the eggs are fertilized, what happens to the parents is irrelevant. It doesn't matter if they live or if they die, the eggs are there and the eggs can be incubated and the eggs will hatch. So like I already mentioned before, we will have control over the development of the eggs. We can already take out eggs that are not fertilized. That's actually a very clever system where at some point the non-fertilized eggs will turn white because there's no fish developing in them, and they will start to degenerate and they will turn white. And then you actually have some machines that will take out that will show egg by egg, and then there's a camera saying, oh, this one's not the right color, and we'll just put some water there and we'll shoot out that one egg. So you can actually sort the eggs based on their colors and take out all the bad ones. So that's a big advantage that we have of eggs being outside of the female. The last advantage that I want to give you is the space effectiveness. You also slightly have this in birds, I think, a little bit from what I've seen. But I think fish and aquaculture takes it a bit further. They live in a water column, and they can actually move up or down as they wish. So that means that they can kind of be stacked. And where you would normally say if we have 5 fish per cubic meter or something, and you have 20 fish, you would need like 4 cubic meters next to each other. That's actually not the case in salmon, because they could also be 4 cubic meters stacked on top of each other. So that means that you just don't need as much space. And here, this is a picture of a hatchery where eggs are being incubated. So you can see here the nice orange color of those eggs being incubated. This is not kept separate per family, but this is a whole bunch of families together. And you can actually see that they have 3 levels on top of each other where those eggs can be kept, can be incubated. So this whole hatchery on a normal day, you can see 4 people here now because they're actually sorting the eggs. On a normal day, this could be manageable with 1 or 2 persons. So that's just really very, very effective. And I think that's a big advantage as well of aquaculture breeding. But we have disadvantages as well, of course. So to go to those right here. Progeny testing, which is something that we really, really would like to do because of the addition in accuracy, is very hard. Because most of the time, these fish have only just hatched. You can still see their yolk sac attached to them. Their parents are probably already dead. So that means for sure, even for shrimp that can have a spawning window of about 3 months during which they stay alive, by the time that you have actual progeny info coming in, their parents are already deceased. It's possible to use cryopreservation for the male sperm, for the milt. The downside of that is that it will severely increase the generation time. So, for example, in Atlantic salmon, if you have a 4 years generation interval, harvest information of the progeny of a sire would only come in after 2.5 years. So that means that rather than after 4 years, you can use this male after 3 years. Sorry, after 4 plus 3 years. So that's use them again after 7 years. So that's a really big increase of your generation interval. And usually the increase in accuracy that you get and thus the additional generation genetic improvement does not make up for the loss in generation interval that you have because of this. So that's a downside that we have. We have a lot of full sub information, but we don't have progeny information. Then the bottom right, phenotyping means to take animals out of the water. That means stress. We literally take these animals out of their element when we need to take a phenotype of them. And they don't like that. Imagine that you will all of a sudden, without warning, be kept underwater. You don't know for how long someone's doing something for you, and then all of a sudden you're taken out again and you can breathe. But no one actually told you to hold your breath for a while, so you're just totally unprepared for that. We try to make this as smooth as possible. The fish are always anesthetized. They have to be, because otherwise we won't be able to get a decent phenotype of them, because they would be moving around all the time. But also because of the animal welfare, we simply cannot take these fish out of the water without them being anesthetized, because it would just be so bad for their welfare. but anesthetizing the whole bunch also causes a bit of stress. We do see some post-handling mortality, as we call it, so some fish dying after we've done a measurement on them. So those things combined mean that we will try to minimize the number of times when we actually do take phenotypes. It differs a little bit per program, but typically for the broodstock, it means that we take information once a year. So for trout, that would be after 1 year and close before maturity. And for Atlantic salmon, that will be at 1, 2, and 3 year and then close to maturity. So that means that we don't have information on like, what does the growth curve look like? Or when fish get a bit sick, how do they recover? Or when they start having deformities, then can we see that coming, or is it just all at once? Well, we can't see it coming, we just see it all at once. So we have no idea how this kind of deformity would develop. And we don't know, for example, if we see an animal that's deformed after 1 year, has it always been deformed or did it slowly happen? So we don't know that kind of things, and that's a downside of simply fish being in the water normally. That you really, yeah, you really notice that in the data that you get. Then something that is salmonid specific again, maturity is the end of life. So this is the picture that I talked about in the background here. You can actually see a fish being stripped, a female. These animals are already dead. They are euthanized using an overdose of anesthetic. And you can actually see the eggs coming out of the female here. So in the top, you put some air in the belly, in the abdominal cavity, and then the air will actually come, the eggs will come out directly into the bucket here. And after this, they will be sent to fertilization. So maturity is sometimes the end of life in salmonids and trout. We can actually use the females for another year, but we really see that extra year, so the third year, that's really hard for them. Not all of them survive it, and the eggs will be of lesser quality, and they will not have as many eggs. So we don't do that for our actual nucleus. We only do that for commercial production. And for maturity to be the end of life, well, like I said, it's very much related to progeny testing is hard. It means that we do not have an opportunity to use a good fish again once it's mature, once it has been spawned or stripped or whatever you want to call it. That's the end of it. If something happens and these eggs will not be fertilized within the next 24 hours, you lose them. They are gone and they will not be able to fertilize anymore. The milk from the males we can actually use for, I think, 7 or 8 days, so we can just make that to the next week. We usually test once a week for which animals are ready to be spawned. So we can only just take that over to the next week, but that's it really. So once they are spawned, that's the end of it. Then for some more downsides of aquaculture breeding. Animals are very small after hatching. You can see this whole bunch of small animals. They all still have their yolk sac intact. These have only just hatched. Good luck keeping those apart. You will not succeed. And that means that they are also too small to give them an individual tag. So usually we will give animals a PIT tag, a passive integrated transponder. It's about the size of a grain of rice for those that have never heard of the term yet. And it will respond to a scanning device. But these animals are actually about the same size of the PIT tag that they will later be given. So it's just impossible to give them any type of identifier at this moment at the size that they are right now. So that means that we have two options. Either we keep the fish separated per family, so that we still know which family they come from, which sire and which dam. right until the moment where we can actually give them this pit tag. To do that, we create a common environment per family. And that has a big downside because you cannot separate good genetics from the common environment that these fish have had. There's some ways around that, but they're not easy. The other option is to pool all the animals to just put all these small little hatchlings together. It's very well possible. And then later in life, after you have pit tagged them, at some point you take a genotype sample, a DNA sample, which we can then genotype to determine their families. Both systems work. Both systems are about equally expensive, at least for like the first 5 to 10 years. I don't exactly know which one would be more expensive on the long term, but and both have their downsides. If you genotype them later, you will not genotype all the animals that you would normally have pit tagged when you keep them in a family unit. So you will have less information, but you will not have the family unit and the common environmental effect that comes with it. So neither of them is ideal, and what you choose depends on the company and the nucleus farm that you actually have. Related to the issue of telling animals apart, these are bigger, they have pit tags, but still, trying to keep them apart from the way that they look is impossible, especially if you think about how they can be together with 10,000 individuals in one cage, that we would call it, or one tank. We cannot record social interactions. I have absolutely no doubt that there will be dominant animals, submissive animals, animals that live higher up or lower down in the water column. Those that live higher up have easier access to food, because if you throw the food, that's where it ends up first. So they might be the bigger breeder. They might also have a bigger effect of light or temperature. So there's a lot of effects that we cannot account for simply because these animals cannot be told apart. And you could say, well, what if you take only 10 and you give them a next donor collar or something? Yes, you could, but the results would be totally different from 10 to 10,000 animals in one tank. So that's not really giving you accurate results. Then on the left, as beautiful as this picture is, because I really like it, it also scares the hell out of me. This is the water source to one of our nucleus farms. And it's the only water source, and we are completely dependent on it. And this is an issue that every nucleus farm, or actually every aquatic farm, doesn't even have to be a nucleus farm, they all have this issue. You are completely dependent on your water source because we need a lot of water. For a nucleus site, that will usually mean to be as close to the source of the water as possible, because that will prevent fish from living upstream that will carry diseases with them. Birds can contaminate this water. There can be pollutants in the water. You don't want that, so you want to be as close to the source as possible. And that means having a farm in often remote areas, and that does cause these very nice pictures to be taken. It also means that you can be hard to reach. Imagine this nice forest being burned to the ground because of a forest fire. Your farm will be gone. There's a lot of water on the farm which you cannot use, because to use the water means to kill your fish. You can actually move the fish because you can't get a truck there in time to move all of them, because there simply aren't trucks to move 10,000 fish in the water while alive, etc., etc. Besides, where would you bring them? Because there's not often a place that has tanks ready for this amount of fish. So it's a huge dependency to have this water that you need to have. And then if you're not a nucleus farm, if you're a production farm, you could be outside in the ocean with your fish, and you will have other reliabilities based on the water that don't happen on the nucleus farm, like the pathogens that can be in there or something like that. That's not to say that the water source might actually disappear all of a sudden. I know there's a farm in Scotland that takes their water from a river, but that's actually a rainwater river. And for that river to exist, there needs to be rain every 24 hours. They do have a backup, and I've never heard of them actually getting into trouble, but it does sound like a pretty risky operation to me. So that's the kind of things that you will hear in aquaculture. So all these advantages and disadvantages, what do they look like in our nucleus, in our breeding stream? It means when we create new families, we will send full sips and half sips to the sentinel cages. That's the commercial situation that we have, and to the challenges for disease challenges to improve the resistance for diseases. Those will give back their info to the nucleus animals that we have, and those will be evaluated using that info and using their own performance. Own performance is absolutely valuable because without it, every animal from the same family would have the same information coming in from the brothers and sisters, and we would not be able to distinct animals within family for the within family selection. After evaluation, we will select our elite broodstock to create the new families again, and those that are not selected to create new families will be used for commercial production. And that's mainly what I wanted to talk to you about, the advantages and disadvantages within the aquaculture breeding. I've also prepared a few slides to tell you about the challenges that aquaculture has as an industry. So that's what I will be showing you now. We are battling a lot of diseases. We have a high disease incidence. This is about commercial stock. The reason is those fish are kept under commercial circumstances. Atlantic salmon will be kept in a cage in the seawater. Water is a perfect way for pathogens to travel. We cannot keep our animals safe from that. There will be wild fish swimming close to these nets, spreading the diseases that they have to our stock. There will be actual sea lions attacking those cages where the fish are, killing the fish, but also spreading the diseases. So we have a lot of issues with that. What follows is sometimes a high mortality and a high antibiotic use to prevent fish from getting sick. Antibiotic use is not something that you want from a sustainability point of view. High mortality is not something that you want from an economic point of view. So we try to improve this. We breed for resistance against multiple diseases simultaneously. If you think of the alphabet with 26 letters, within Atlantic salmon, the most important diseases right now are BKD, SRS, IPN, and Caligus. In trout, we have bacterial cold water disease. We have IH and V. In shrimp, we have WSSV, CEMS. I don't even know what all those abbreviations mean, really, but I know that they are all diseases that we try to eradicate by improving the resistance of our fish against those diseases. And it's not easy improving all of them at the same time because some of them just ask for a whole different way of fighting this disease by the fish than others. And the fish cannot excel at all of them at the same time, especially if you also want it to grow at the same time. One special case that we had in Atlantic salmon was IPN, infectious pancreatic necrosis. We hit the jackpot. In 2008, we discovered that there's two specific genes explaining 99% of the resistance against this disease. We can explain them using 5 QTLs because we haven't actually found the specific genes, but we've got QTLs explaining them. And it's working perfectly. We genotype animals on 5 SNPs, and we know how resistant they are against the disease. Problem is, everyone wants miracles right now. Finding these QTLs has led to a widespread belief in the industry that genomics is the way forward, QTLs are the way forward, and people expect people with a bit less knowledge on genetics and genomics, they think that we can find a QTL whenever something happens, which is sometimes difficult to explain that that's actually not the case for most things that we encounter. So that's the downside of it, but we did find one jackpot, so that's nice. Varying circumstances. I said before that we have nice high heritabilities. That's true under standardized situations. So it means that in those standardized situations, we are very well able to improve our genetics. The problem is there's big differences between farms and between years. One farm might keep their shrimp in a nice industrialized pond, and the next is just a small local farmer that has a muddy pond in his backyard where he will throw in the shrimp and have a look at them a few months later. That's a big difference. We can have differences between years. We can have algae blooms in the seawater, and that means that there's a lot of toxic material coming your way all of a sudden. This can happen within 2 weeks' time, and they will kill you. Well, 25 to 95% of your stock, or maybe even 100% within a few days, without a warning. So as soon as farmers start hearing, hey, there's algae bloom and my fish are dying because of that, if they have fish close to harvest weight, they will immediately start taking out their fish. And that means that we have variation in the size that the fish are at the moment that we start harvesting them. So that leads to a varying performance that we can have, where we have information on, and also the performance that the farmers see, because they will actually expect a certain performance from the stock, but the circumstances will make it almost impossible to get that. So that means that we're just trying to breed animals that can basically handle everything, which is quite impossible, but we need to aim for that anyway. Finally, and this is my last slide, we have a need for sterility. Once animals start maturing, that's bad for the flesh quality, because they will start to take the fats and they will start to take the colors out of their flesh, in case of the females to produce the eggs, and in case of the males to produce the nice phenotypes, the nice looks, the bright colors on their skin to show that they are actually mature. And that causes bad flesh quality and thus less value for what you have in your cage. We also have the problem of mature animals that can reproduce. In the case of shrimp and other fish that are kept in third world countries, we actually have an issue that people will try to set up their own local breeding program. And they will say, hey, I know my neighbor has some nice fish or shrimp from Hendrix Genetics and they do pretty well. Let's just go in tonight and steal some and try to make our own breeding program. That happens. It's very difficult to prove. It's very difficult to prevent. So we want animals that do not reproduce, so they actually don't give away, so to say, your genetics and your research. Last but not least, we have to take into account the environment. We have SKPs. SKPs means fish or shrimp or any animal that escapes the place where it's kept. So in the case of Atlantic salmon, it might be that a cage is broken because of a seal attack. I even once saw a picture of a tuna that broke a cage and just caused havoc in there. Shrimp are often kept very close to their wild counterparts in the mangroves, and if someone accidentally leaves a valve open, they will just crawl out and go into the wild. That's not good for the wild animals that are there, because they might have a disadvantage, or actually the escapees might not be as good, but they will still reproduce with the wild animals, and that will cause problems for the wild populations actually. So that's also something that we want to prevent. And with that, I come to my closing slides. I want to thank you all for your attention. And I hope that you enjoyed and that I was able to teach you something today.
Great, thank you, Vera. Does anyone have any questions? Feel free to throw your hands up and turn your cameras on as well. Not seeing any initial questions. I might ask. Oh, yes. Is that you, Flavio?
Yes, yes. I can start with one clarification, but first I'd like to thank Vera very much for this nice presentation. It was really the clearest presentation that I attended in terms of
Thank you.
showing the differences of aquaculture and land animals in terms of the structure of the breeding program. Was really, really nice.
Thank you.
So thanks for that. But I have a question, it's not really a question, clarification. You mentioned that the traits, even for the health traits that you work with, the heritability is kind of higher than usually we see for livestock species like not like land animals. Uh, and just to clarify, but the heritability that you estimate there are heritability of the means of the families, right?
What do you mean the heritability means?
Is the heritability of the mean value of the families is based
Yes,
on
that's true.
family heritability, right? So if you do the same with the animals, you would have a higher heritability, right?
What we actually work with is the days to death. We are globally, everyone is trying to find an alternative, but the way that disease challenges happen currently is mostly with infectious diseases, that animals get infected and we check how long it takes for them until they die without any treatment. And we note down the days to death from infection to mortality for each of the animals. So we actually do know the variation within the family. We also know which of the animals survived because we stopped the challenge at a certain point. So we do have within family variation.
Yes, yes, understood that. I'm just saying that when you look at the heritability that you are showing that are higher,
Mm-hmm.
high, because those heritabilities are the heritability of the family means, right? It's not the heritability of a single individual. If you measure a single individual,
No. It's the heritability overall for the population. Yes.
yeah. So what I'm saying that heritabilities in aquaculture usually they look higher
Mm-hmm.
because they are based on family means, right? Because you do family selection.
We also have within family selection, both within and between family selection.
I'm talking about the parameter heritability there. I like to put in the context the heritability that you showed to us and the heritability that you have in land species. Usually,
Yes.
the land species we calculate heritability based on a single phenotype of an individual,
Mm-hmm.
not the average phenotype of a family. Right? In
Yes.
aquaculture, usually the heritability is based on a family, right? The record.
No, it's not based on a family. We have a family structure, but we have information on each individual within the family.
Oh, so the heritability that you presented then is based on phenotypes of single individuals.
Yes.
Okay, so that's what I like to clarify. So then the heritabilities are really higher.
Yes.
Okay.
Yes, they are generally quite high compared to terrestrial animals.
Yeah. Okay, so yeah, that's important.
No, it's an important part indeed to clarify that, but it is based on individual information. Yes.
Okay. Yeah. Because but even though the ability that you are more interested when you do family selections is the heritability of the family average, right?
We try
That's
to
the
take
heritability that's relevant for you.
no, we try to take as many individual data as we can possibly get. So we work with very few averages for families.
Okay, but you told me that in British scheme you don't have a progeny test, right?
No, but we do have full SIPs that we can use. And we have an own performance which is correlated to the trait that we measure in the full SIPs. And we
Yeah,
also use genomics for to get more information.
and for the full SIP, you are able to measure the individuals, each full SIP or the full SIP as a group.
Yes. Each full
Individually?
SIP. No, each full SIP individually.
Individually. Okay. Okay, that's very interesting. That you can do that. And you do for all species, even for shrimp?
Yes. For shrimp, if we cannot identify individuals, we will use genotyping to make sure that we know the parentage.
Okay. Okay. Thanks, Vera. Thanks for clarifying that.
You're welcome. Thanks for the question.
Thanks, Flavio. Elizabeth?
Yes, can you hear me, Vera?
Yes, I can.
That
Hi.
was a very nice talk, and you covered a lot of ground on salmon and taught me a lot of things that I don't know. But I did find something particularly fascinating, and that was, first of all, just a quick question about SNP density on your chip. What are you finding you have to use? Like, how many SNPs do your molecular people use? Do you know off the top of your head?
I know, to be honest, I'm not sure how much information I'm allowed to give there because we have our own SNPs that we use that are really used only within the company. So I don't actually know how much information I can give.
No, you can't. You shouldn't tell me your SNPs, but it might be okay to tell me how many you use.
Yeah, so usually we have about 16 to 50K SNPs.
Yes, okay.
It depends a little bit on the species and on the line that you're working with.
So yes, the trouble we had when I was doing some work on this is 50K is still pretty expensive for the amount, the price of the fish.
Yeah.
So are you aware of how many get genotyped at 50K, 10 to 50K?
No, that I do not specifically know. I think the 50K is mostly used for research purposes.
But say the 10K or whatever, would you do like all the candidates?
We do genotype all the candidates, yes. There will be more than a 10K SNP, but not as much as 50K, so that would be like 20K or something.
Because what I find is you have all these progeny, but the minute you start using genomics, which you have to do because so many of the traits you can't directly measure, like you can get their freshwater growth if you keep them in fresh water, but that's all you can get up pretty much if you're doing disease challenges. Then you have the problem, your intensity of selection is not really that high because you can only genotype so many candidates, right?
Well, we do have very good correlations between freshwater growth and seawater growth.
Oh.
So those correlations can be, normally they will be like 0.6. They can be up to 0.8 actually sometimes. For specific populations. So that actually means that we can indeed use their own performance to get a relatively good estimate of their seawater performance would be.
Right, and you do genotype on some kind of chip all the candidates, but wouldn't your intensity of selection be how many candidates you genotype versus how many you use? I mean, I'm not specifically an animal breeder, but
there's debate about that, yes. So you could say like, well, we start out with 15,000 per family, so that's in the end, like if we use 10, that's a very high selection intensity. You can say, well, we genotype 1,000 in total and we use 500 of those, so then what's your selection intensity? It's debatable which one is correct. I tend to agree with you that we should only look at the ones that are in the end actual candidates, but we do have pre-selection before that that I wouldn't immediately say is just random. So I can't say that that's no selection intensity. I can't say that it's high selection intensity. So it's debatable.
But then my other question, and this is again an animal breeding question, you're breeding for disease resistance, which from everything I've heard is absolutely critical. But you already talked about how no fish is a superstar on its own, that the ones that are most resistant means they're putting a lot of energy into their immune system and maybe not so much into growth.
Mhm.
So do you have fast growth lines? And then as some companies that I've heard of have say a BKD resistant line or
Yeah. We
IPM
do.
resistant.
Yeah, it's not always a whole line that is more resistant, but we can sometimes select some animals and create some families that are more resistant with the number of eggs they create. That's easily enough to produce enough fish or shrimp or whatever for a certain number of customer. But sometimes indeed in shrimp we do have real lines that are fast grower or that are more resistant or stuff like that. Yes.
Okay, that's really fascinating. Thank you. I really enjoyed that.
You're welcome. Thank you.
Thanks for the questions, Elizabeth. Christiana?
Hi, Vera. Thank you for the great presentation. I work on dairy cattle where I study inbreeding, and I was wondering what inbreeding look like in aquaculture. Like, is it an issue, and how do you monitor it?
Generally, we don't have a lot of issues with that because the domestication has been so recent. We do monitor it. We monitor it on a pedigree basis, but also on a genomics level. We try to keep it under control. But I've never seen it being a problem in our lines. And the advantage that we actually have, if it is a problem, because domestication is so recent, we can actually sometimes, not always, take wild animals and add those to the program to reduce the issue.
Okay, thank you.
You're welcome.
Thank you, Christiana. Does anyone else have a question? I'll ask a quick question while we're waiting. You mentioned that, I can't remember for which specific species, but you gains of 0.5 kilo a generation. Is there any thought or any worries about speeding up maturity too fast? The thing that comes to mind for me is with chickens, that they grew too fast, that their organs couldn't sustain. Is there thoughts of that with aquaculture? Or
Yes, that definitely is. And this is where we really can learn from the other species that have been domesticated so long ago and where the whole process of genomic improvement started so long before aquaculture. We know that we may involuntarily select for one thing while selecting for another. So especially maturation, what you're mentioning is a very good one. We know that the faster growers, the one that I was talking about is Atlantic salmon. We know that the faster growers are also the ones that tend to mature early. There's a very high correlation between that. So we make sure to monitor both those traits and to improve one while not improving the incidence of the other. Yes.
Perfect. Any last questions for Vera? Okay, I'm not seeing any, and since we're right on the 7:30 mark there now, I'd just like to thank you again, Vera, for the great talk and great explanation to explain to a lot of us more terrestrial animals, as you put it. Definitely gave me a better understanding of aquaculture. So I really, again, thank you for the great talk. And thank you
You're
for
welcome.
everyone that was here today. We'll see you all next week.
Okay, thank you very much.